Hello. Today's unit, we will be talking about the physiology of digestion. And again, this will kind of lay the framework for us as we move forward and learn about metabolism as well. So again, this lecture is entitled Ingestion to Energy Metabolism. So again, when we think about this process of digestion, again, it's essential in order for us to be able to bring food in. We're going to break it down into smaller components. The next thing we'll do is have absorption occur, which is the movement of those food molecules from the digestive tract into the bloodstream. And then the next process is the actual metabolic process, where we're able to use those molecules as ATP, which is our most basic energy unit. So again, when we think about what's the general, goal of digestion, we want to break down those ingested food molecules through enzymatic and mechanical action so that they can be absorbed in the body. So a nutrient isn't actually technically in the body until we've absorbed it. And again, the process of absorption involves us moving that food molecule from the digestive tract into our bloodstream. Okay. Two different types of digestion need to occur in order for us to eventually absorb those molecules. The first is mechanical digestion. And mechanical digestion involves the breakdown of large pieces into smaller pieces without altering the chemical configuration of that food molecule. And then chemical digestion involves breaking food into simpler chemicals. Okay, so again, we need both mechanical and chemical digestion to occur in order for us to eventually absorb those molecules. Here you can kind of see the path that food will flow through. In addition, you can also see the accessory organs listed on this picture, as well as kind of their general goals. Okay, so again, we'll go through these in significantly more detail, but this just kind of gives you an overview. Food will flow through the mouth, through the esophagus, into the stomach. Next, it'll move into the small intestines, into the large intestines, and then waste products will be removed through the rectum. Okay. Additionally, we have many accessory organs, and the job of an accessory organ is to assist in the process of digestion, but it doesn't necessarily serve as a pathway for the food to flow through. Okay. So we have salivary glands, we have the liver, the gallbladder, pancreas. All of those will assist in some sort of process as it relates to digestion, but they don't actually serve as a tube through which the food will flow. Okay, so here you can see the flow of food through the digestive tract. Again, pay attention to my arrows. They're a little bit like light. So we're going to move from the mouth to the esophagus to the stomach. Then notice that we're going to the duodenum, jejunum, ileum, into the large intestine or the colon. And then finally, any waste products will be removed via the anus. So again, that's the tract. When we think about the digestive tract or the GI tract, this is the flow of food through that particular system. This I just think is interesting. So this just kind of shows us on average how long food would spend in each of those locations. So again, in the mouth, about 1 minute, unless you're like my yellow lab, and then it's like 0 seconds in the mouth. He like engulfs food and he like chugs it, which is hilarious to see him eat his dinner in like 10 seconds if you let him. But the average person should spend about 1 minute with food in their mouth. Then it'll be traveled through the esophagus, and again, 4 to 8 seconds here. Then in the stomach, it'll spend 2 to 4 hours. Again, the length of time spent in the stomach depends on the composition of that food. So again, if it's high in proteins and fats, then it might spend closer to 4 hours. If it's primarily simple sugars, something like Skittles or Pop-Tart or something like that, then maybe closer to just the 2-hour mark. Next, we'll move into the small intestines, where it can spend 3 to 5 hours, and then in the large intestines or the colon for 10 hours up to several days before it's eventually eliminated. Okay. As I mentioned, we have these accessory organs. that are going to assist us with the process of digestion. However, they are not part of the actual GI tract. So salivary glands, liver, which you can see in this picture, gallbladder is here, and pancreas is located here as well. Okay, so let's take a look at those accessory organs first. Okay, so salivary glands are going to help us to do a couple different things. Number one, it's going to allow us to secrete saliva, and saliva includes salivary amylase and mucus both. The goals of saliva in general are to help us to moisten those food particles. And again, if you remember back to the sensory physiology unit when we talked about taste, it's important that the food molecules are dissolved in a fluid or a liquid of some sort in order for us to be able to actually receive the taste of those items. So saliva will help us with that. It can also help us with the beginning of chemical digestion of carbohydrates, and that will be done through the work of salivary amylase. Okay, so salivary amylase is going to help us begin the process of chemical digestion of carbohydrates. And again, that will occur within the mouth. And then also we have mucus within the saliva or coming from those salivary glands. And this is going to bind to food particles and help us to lubricate that food as we swallow it. Again, imagine eating something kind of sharp like Doritos, for example. This mucus is super important so that we don't suffer any abrasion as we move those like sharp edges down the esophagus. So the mucus will kind of help soften and lubricate that like little chunk of Doritos that we've consumed in order to prevent any type of injury to the esophagus. Okay. The liver is freaking cool. The liver can do a bunch of different things. It's going to help us to destroy toxins such as the toxins found in alcohol, for example. It's also going to secrete bile, and bile is going to be stored in the gallbladder, which you can kind of see here. Bile is going to help us with fat breakdown. It's also going to serve as a processing site for many of our almost all of our nutrients. So the nutrients, once they're absorbed, can be carried to the liver where they can be processed or reconfigured into some sort of other alternative form. Okay. Okay. As I previously alluded to, the gallbladder will serve as a storage pocket, basically, and all it really does is it stores bile. Okay. And bile is going to be released into the small intestines and used to help us to break down fat molecules. So that's really all the gallbladder does, is it just holds this substance known as bile. It is actually green like this. And like I said, that bile will be released into the small intestine so that we can use it to kind of like break down fat molecules in that location. Okay. Pancreas does a bunch of different stuff. It creates several different enzymes, or I have it listed as juices here, that are going to aid in digestion of carbs, fats, nucleic acids, and proteins. Pancreas also serves as the location for production of insulin and glucagon as well. Okay. Insulin and glucagon will help us to monitor blood glucose levels. So again, when we think about digestion, I've kind of broken this lecture down into 3 separate areas. So I want to take a look at what happens when we digest carbohydrates, when we digest fats, and when we digest protein. So we're going to look at each of these nutrients kind of as a separate entity. However, I do want you to keep in mind that in general, like in our normal lives, we're consuming all 3 of those nutrients at the same time. Okay, so for example, I just had lunch. I had spinach salad with some tomatoes and avocado, and then I had like some pasta salad on top of that. So the pasta salad was like this bacon ranch pasta salad. So again, think about the composition of that meal. A lot of carbs, some proteins in there, and also some fats as well. So again, not just simply consuming carbohydrates. However, we're going to kind of break these up so that we can better understand what's going on. But keep in mind and do appreciate that all 3 of these processes can occur simultaneously. In order for us to understand what's going on with carbohydrate breakdown, we need to understand how carbohydrates are organized to start with, okay? So a polysaccharide is several sugar molecules that are going to be linked together. Polysaccharides can be known as starches or glycogens, and they're made of 11 or more simple sugars that are linked together, okay? The simple sugars are known as monosaccharide, mono meaning one, saccharide meaning sugar, so it's one simple sugar molecule. And these sugar molecules might include one of three things. So that we have three different types of monosaccharides. We have glucose, fructose, and galactose. So those are your monosaccharides. We can link them together in twos. Di means two, saccharide, sugar. So I've got two sugars linked together. And there are three different types of disaccharides. So I have sucrose, which is composed of one fructose molecule and one glucose molecule. I have lactose, and lactose is made of galactose and a glucose molecule. And lastly, I have maltose, and maltose is consistent of two glucose molecules linked together, okay? Let's see, what else should I tell you? So in general, like we're consuming carbs in the form of a polysaccharide, and we want to break them down into monosaccharides, okay? Once we break them down into monosaccharides, we're ready to absorb them and get them into the bloodstream. If we want to then store those sugars inside of our body, we go back in the reverse way. So we will build those back up and store them as polysaccharides, okay? So they're consumed as polysaccharides. We break them down into monosaccharides in order for us to absorb them. If we decide to store them within our own bodies, we are going to convert them from monosaccharides into polysaccharides. Okay. So let's take a look at what's happening within each of those digestive organs along the GI tract. Okay. So first thing we're going to do is we're going to put those carbs in our mouth, and we'll start some mechanical breakdown through the chewing of those particular items, okay? As you already know, we're also going to release salivary amylase, and salivary amylase is going to begin the breakdown of those carbohydrates from a chemical standpoint as well. Okay. Then those food molecules, again, will spend about 1 minute in the mouth, are going to move through the esophagus, and this will simply serve as a transit from the mouth to the stomach. Again, only like a couple seconds in the esophagus. Salivary amylase is continuing to work as we move that food from the mouth to the stomach. Once we reach the stomach, we're going to release a molecule known as hydrochloric acid. And hydrochloric acid actually stops the digestion of carbohydrates. Okay, so we're going to shut off the work of salivary amylase, and so carbohydrate chemical digestion is not going to happen within the stomach. We will have mechanical breakdown occurring within the stomach via churning of the smooth muscle. And so again, I want you to imagine, like, if you're familiar with like baking a bread, if you were to knead the bread, you're kind of like pushing the bread around. That's kind of what happens in the stomach with this whole process of churning. Okay, so again, imagine your stomach is like this. It's going to kind of like mush the food together basically, and that's going to help us to mechanically separate some of those particles. Let's see, what else do I want to tell you? Yeah, so the hydrochloric acid denatures the salivary amylase. So it basically shuts off that salivary amylase. And the food at this point within the stomach becomes a bolus. And I believe once it's a bolus, it's just a different term. But it's basically like, because you smushed it all together, now it's like a big conglomeration of food. Okay. So next thing we're going to do, we're going to move that bolus of food into the small intestines. And the first place we'll go is the duodenum. That's the first section of the small intestines. Again, movement into the small intestines will spend about 1 to 4 hours here if we're a carbohydrate. The majority of digestion and absorption will occur in the small intestines for carbohydrates. Okay, so let's take a look at what's going on here. So the duodenum, the pancreas is going to release pancreatic amylase, which you can see here. That's going to help us to take starches or those polysaccharides, so long chain carbohydrate molecules, and snip them off into disaccharides. So disaccharide, again, we said were 2 sugar molecules linked together. Okay, so pancreatic amylase is responsible for breaking down polysaccharides into disaccharides. Okay. Next, we have these 3 enzymes, lactase, sucrase, and maltase. And they're going to take their disaccharides and break them down into monosaccharides. Okay, so again, sucrase will take our sucrose disaccharide and break it into a fructose and galactose. Lactase is an enzyme that will take our lactose disaccharide and break it into its simpler form of 1 glucose and 1 galactose molecule. And maltase will take our maltose disaccharide and break it down into 2 simple glucose molecules. Okay. Let's see, what else do I want to tell you? In order for the carbohydrates to be absorbed, they need to be in the form of a monosaccharide. Okay. Within the ileum and jejunum, we'll also see absorption occurring. And again, absorption involves the movement of those monosaccharides from inside the small intestines to into the bloodstream. And we'll talk about this coming up, but there are 2 different ways in which we can absorb them. We can absorb molecules through the process of active transport, and we can absorb some molecules through the process of facilitated diffusion. Any unabsorbed or undigested polysaccharides or carbohydrates will pass through the large intestines and they will be eliminated. Again, if I'm consuming some sort of fiber, for example, I gave the example that I had like some spinach salad and some pasta salad as well for lunch today. A fair amount of fiber found in, spinach and potentially in the pasta salad, depending on like what type of noodles I had, if I had like whole wheat noodles. But that fiber and probably some of those undigested polysaccharides will pass on to the large intestines. Keep in mind, we're actually going to absorb, like it's over 90% of the calories that we're consuming, particularly carbohydrates. Those are one of the easier ones for us to break down and absorb. So again, we talked about, in general, the process of absorption involves the movement of these food molecules from inside the digestive tract to into the bloodstream. Okay, and again, 2 different things we can do. We can do facilitated diffusion and active transport. Okay, so facilitated diffusion will allow us to move fructose, and active transport will allow us to move glucose and galactose. So let's take a look at what those mean, right? So facilitated diffusion, again, this is how fructose is going to move. And basically what happens is, imagine that this blue part out here is inside the small intestines, and imagine this yellow section down here. Sorry that these are a little bit blurry. This yellow section down here is where the blood supply is. Okay, so again, my goal is to move these molecules from the blue area into the yellow area. And that's what absorption is, movement from small intestines into the bloodstream. Okay, so I'm going to use this kind of like lock and key mechanism. Okay, 1 fructose molecule comes in and binds to this selective carrier. Then the carrier flips over and dumps that molecule out onto the other side. Okay, so that's what facilitated diffusion is. This is very slow, right? So imagine that I can only move 1 molecule at a time. right? So I put one in here, it flips over and dumps it out. And then this carrier has to turn itself back over so that it gets in this configuration so that another fructose molecule can come in, bind. Now I flip it over and dump it back out. Okay, so fructose is the slowest of the monosaccharides to be absorbed. We would certainly want to avoid this type of sugar if we were in a hurry to get sugars into our system. So again, if I'm working with a diabetic who is low in blood sugars, I would want to avoid fructose molecules because these are slow to get in. I would want to choose something that was maybe higher in glucose and galactose instead, because those absorption mechanisms are going to be a little bit faster. Okay. Active transport. Again, sorry that my image is a little bit blurry. The key things to note with active transport, I need an energy requirement, or I have an energy requirement in order to allow this movement to occur. Okay, so the energy is in the form of ATP, so I have to have an investment going on here. I'm also going to move the molecules from low concentration to high concentration, so they're kind of like going upstream, basically. Again, because we're going against the concentration gradient, it will require some sort of energy contribution. These active transport channels, again, notice that it's just like I can flow many molecules through pretty fast. So it is significantly faster than the absorptive process of facilitated diffusion that we just talked about. Active transport will also be used to carry sodium, for example, some minerals and some amino acids as well. So active transport is used for a variety of reasons within the absorption process. Here you can see a glucose sodium SYMPORTER. This I think is really neat. So basically what a SYMPORTER is, a SYMPORTER means that I'm going to move two molecules in the same direction. In this particular case, we're going to move both carbohydrate and sodium at the same time. Okay, so I'm going to move a salt and a sugar at the same time. Okay, so again, instead of needing ATP as my fuel source, I actually use the gradient of salt in order to allow those sugar molecules to also move. Okay, so again, if I have one sodium and one glucose molecule, I can move them across the membrane together without needing an energy input from ATP, which is nice. So that's a glucose sodium SYMPORTER. Again, when we think about getting sugars fast, something like Gatorade, for example, where I'm interested in getting sugars into my bloodstream right away, contains sodium. And this is why something like Gatorade has sodium in it, because I can move sodium and glucose together. That's going to facilitate faster absorption of those sugars. So I'm able to use those sugar molecules faster than maybe a product that didn't contain salt. Okay. So again, as we said, we will have some absorption occurring within the small intestines, facilitated diffusion if I am fructose, and active transport if I am glucose or galactose. Okay, so I'm moving those molecules from inside of the GI tract into the blood vessel. And the blood vessel that I'm going to utilize is known as the hepatic portal system. And the hepatic portal system is going to carry those nutrients from the small intestines to the liver. Okay, so hepatic portal system is a system of capillaries that's going to, or blood vessels, we should say, carry nutrients from the small intestines into the liver. Once all those sugars are going to reach the liver, all the sugars are going to be converted into glucose. Okay, so I'll take those fructose and galactose molecules and I'm going to convert them into glucose molecules. Then glucose can do a couple different things. It can be released back into the blood if I need more sugars in my blood, or it can be stored in the form of glycogen. Okay, it can also be stored in the form of fat as well. If I overconsume carbohydrates, I certainly can convert those into fat molecules as well. So those are all the different possibilities that we see with glucose once all the sugars are converted into such within the liver. If this is not helpful for you, that's okay. Just ignore it. Like I have told you in the past, I'm kind of a visual learner. So I like to see, you know, different images. That's helpful for me. But here you can see in the mouth, we've got salivary amylase, and that's starting to break down those starches. Again, in the stomach, no carbohydrate digestion is occurring. In the small intestines, again, I have pancreatic amylase, which we talked about is going to break down starches into smaller molecules such as our disaccharides. Again, here you can see those 3 enzymes that are going to break down our disaccharides into their appropriate monosaccharides. So again, maltose will be broken down into 2 glucose molecules. Lactose will be broken down into glucose and galactose, and sucrose will be broken down into glucose and fructose. Again, nothing new on this slide, just kind of shows you how we consume these nutrients and what's going to happen to them to get them into the bloodstream. Okay, so here you can see dietary carbohydrates in the form of polysaccharides, or those long chain molecules, disaccharides, which are the 2 chained sugars, and then monosaccharides. Okay, again, you can see salivary amylase playing a role in the mouth. Pancreatic amylase will play a role in the small intestines until eventually we get our 3 monosaccharides, which will be carried in the hepatic portal system to the liver so that they can all be converted into glucose. A little bit of more information about what the hepatic portal system looks like. Again, here you can see the small intestines, and we're breaking down those foods. We're ready to absorb them. So those food particles are going to move from the small intestines into this hepatic portal system. The hepatic portal system will then carry those food molecules to the liver where they can be processed. Once they're processed in the liver, they will travel to the heart. And then the heart will utilize its pump mechanism in order for it to send those nutrients to all of the body. Okay. Yeah. So kind of defining some of these terms here. Hepatic means liver. Portal means 2 capillary beds. So again, I have a capillary bed at the level of the small intestine so that I can pick up those particles, and then I also have a capillary bed at the level of the liver, and that's going to allow me to drop off those nutrients to the liver so that the liver can process those. Okay, so that is the nitty-gritties of carbohydrate digestion. The next nutrient that we'll look at is fats, and we'll take a look at how fats are digested and absorbed. So first thing we need to understand is the kind of basic structure of a fat or a lipid. So fats are water insoluble. Basically that means that they don't dissolve in water. They don't like to dissolve in water. If you were to put oil in a pan with water, you would see the oil separate from the water because they don't like to be in there. They're water fearing or hydrophobic in nature. Okay. Lipids are consumed as triglyceride, tri meaning 3, and then we have a glycerol backbone. So we have your glycerol backbone here, and then we have 3 fatty acid tails, which can be seen here. Let's see. When we break down triglycerides, they're going to be broken down into long, medium, and short chain fatty acids and then absorbed. We can use them for fuels, or we can convert them back into triglycerides so that they can be stored. So again, we consume it in this form. In order for us to absorb it, it has to be chopped off of the glycerol backbone and made into short, medium, or long chains here. And then those chains can be absorbed. Once the chains are absorbed, and the glycerol will be absorbed as well, we can convert it back into this form if we want to store it. or we can just use it for a fuel. We can use those fatty acid chains as a fuel. So those are kind of the fates that we're looking at with our fats, okay? So again, imagine that avocado, which was part of the lunch that I consumed today, fat in the mouth, little digestion is occurring there, maybe a little bit of mechanical breakdown, but no chemical breakdown occurring. Move through the esophagus into the stomach, and in the stomach, I'm going to release something known as gastric lipase. And gastric lipase is going to break down those triglycerides into a diglyceride and a fatty acid tail, or a free fatty acid. Let's see if I can draw something like this for you. So again, there's my triglyceride. Again, I have a glycerol backbone and 3 fatty acid tails. Once I have gastric lipase in the situation, I'm going to convert this into that and that, okay? So I have a diglyceride, so I have 2 fatty acid tails and 1 free-floating fatty acid, okay? So that's what I did with this molecule of gastric lipase. Also within the stomach, we'll have some churning occurring, and this will help us to break apart large pieces of fat. Fat will spend about 2 to 4 hours in the stomach, and 1/3 of those triglycerides are broken into these free fatty acids, or these fatty acid tails. Okay. Next, we'll move to the small intestines, and in the small intestines, we have fat that. Once fat enters into the small intestines, we're going to release a hormone known as CCK, which you can see here. And CCK is going to message the gallbladder and tell the gallbladder to begin releasing bile. Okay? And bile is going to help us to further break down fats. Pancreatic lipase will finish the digestion of fats, breaking it down into glycerol and short and medium chain fatty acids. So again, imagine I had this diglyceride. Through the help of pancreatic amylase and bile, I'm able to have the glycerol backbone, and then maybe I have 1 long chain fatty acid, 3 short chain fatty acids, and 2 medium chain, or whatever, okay? So as long as they're off this glycerol backbone, they're good to go for absorption. So we can absorb long chain, we can absorb short chain, and we can absorb medium chain. Okay, so any of those are possible. Okay. So again, short and medium chain are going to absorb through that intestinal wall via passive diffusion. Okay, so they're going to travel into the hepatic portal system, just like we talked about with carbohydrates, and they're going to go to the liver where they can be processed. Okay, simple enough. Passive diffusion is nice. It doesn't cost us anything. They're just going to travel across the membrane wall, no problem. Right? Long chain fatty acids are going to form what's known as a micelle, and a micelle is a tiny bubble that's going to carry the fats across the membrane. Instead of heading into the hepatic portal system, however, they're going to enter the circulation eventually through the lymphatic system, okay? So these long chain fatty acids are going to get in like this little backpack, which is a micelle, and they're going to be dropped into the lymphatic system. And so now I've got these long chain fatty acids floating around in my circulation, because eventually this will dump into my blood and travel through my heart. Again, you can see why long chain fatty acids might be problematic for us. We don't want a whole bunch of long chain fatty acids floating through our blood, because we don't want our blood to be fatty. Again, the reasons why it's bad to have fatty molecules, or a lot of fatty molecules within the blood, is because this could lead to clotting and other types of issues, stroke, coronary artery disease, atherosclerosis. Those are all examples of diseases that can occur if I have too many long chain fatty acids. They could potentially cause a blockage, like mentioned, or build up on the walls of the vessel, which is also not good. Okay. So again, another summary here. So short and medium chain are going to be carried to the bloodstream, that hepatic portal pathway, and then they'll be processed in the liver, which is all nice and fine. Those long chains are going to combine with a protein carrier and form a lipoprotein. So again, imagine that the protein is like his escort, basically. And once they have their little escort, this protein escort, they are known as chylomicrons. As we mentioned, those chylomicrons are going to be carried into the lymphatic system and then released directly into the blood. And then they can be delivered throughout the body without any processing. Okay. Again, it is problematic to have a lot of these long chain fatty acids because they're not processed. Okay, so now we've got a bunch of long chunks of fat floating around through the blood. We don't want that. Okay. Once we have the fat in the blood via either of those absorptive processes, the long chain heading in through the lymphatic system, and the short and medium chain heading into the hepatic portal pathway via passive diffusion, once they do that, they can be used for a couple things. We can use it for energy, or we can store it as a triglyceride or store it as a fat molecule. Those are your two options as it's associated with fats. One other thing to note on this slide, we have lipoprotein lipase, which is an enzyme needed to break down the triglyceride into a glycerol and fatty acid. And this will occur if I want to use that fat as an energy source. So people that are good fat burners, such as endurance athletes, for example, will have high levels of lipoprotein lipase in their capillaries and their muscles, which would enhance my ability to be a good fat burner or be good at being able to use fats as a fuel source. Okay. Here you can see the relationship between like these micelles and chylomicrons. As far as I'm concerned, you can use those words interchangeably, but basically you see the fat here, you can see the bile causing fat digestion. Again, those long chain fatty acids will bind and create what's known as a micelle. Once they cross over into the lymphatic system, they become chylomicrons, but it's basically like the same thing as far as I care about. And then they'll enter into the lymphatic system where they eventually dump into the blood. Okay. And then our last of the macronutrients to look at as it relates to digestion is proteins. Again, proteins aren't really a huge contributor for us as it relates to energy needs. However, they're essential for us in providing structure to the body. They're also a key ingredient in producing enzymes. So even though they're not huge as it relates to providing us energy, they do a lot of other things that are very important for our basic physiological functioning. So in the mouth, nothing going on. Proteins are not undergoing any digestion. In the stomach, protein digestion begins to occur. We're going to have what's known as denaturation of the proteins. Okay, and denaturation is a process by which a protein begins to unravel. I have an image here to show you of how this looks. Okay, so when we consume proteins, basically what we're consuming is a strand of amino acids, and there are 20 different amino acids. So imagine that there are 20 different options for beads, and I can arrange these beads in any sequence I want. Okay, so I can have, 4 red beads, and then I have 2 yellow and 1 green, etc., etc. I could have all red, I could have all green. So again, there's tons, just thousands of different ways in which we can organize these amino acids on this strand of beads. Okay, so that's your primary basic protein structure. Now, basically what I'm going to do with that long strand of beads is I'm going to kind of like twist it up and twirl it into this little ball. And this is what it's going to look like when I consume it. It's like a little ball of twisted beads, basically, on a strand, okay? So denaturation involves me like straightening out that strand of beads, okay? So denaturation is the process by which the protein begins to unravel. Again, we're not actually breaking anything down. We're just like unraveling this protein strand. And the reason that that's important is because it allows these little chemical bonds or these little connections between each of the beads to become available to the enzymes for digestive processes, okay? So first thing I have to do is unwind this or denaturate it in order for it to be available for digestive processes, okay? So that's what denaturation is. Denaturation occurs in the stomach with the help of hydrochloric acid, okay? We also have pepsin within the stomach. Pepsin is going to help us to start to break down those long beaded chains. And this is responsible for 10 to 20% of protein digestion, and it helps us to break those strands into shorter chains of amino acids, okay? As we move into the small intestines, again, the majority of protein digestion will occur in the small intestines. Two more enzymes that are of importance to us. We have proteases, and proteases are enzymes that are going to break down the protein into even smaller components. These proteases are made in the pancreas and small intestines. And then we also have peptidases, which are going to break those short protein chains into lengths of 3 amino acids or less. Okay, so they're kind of like the smallest, or they're going to be working with the shortest chains of those amino acids. Okay. Again, we talked about this in the process of denaturation. I've got this strand of beads that's all kind of smushed together, and I need to unravel that strand so that we can see those primary structures so that each of those chemical bonds are available for those enzymes to go ahead and get to work on. Okay. Process of protein absorption. As I mentioned, there's 20 different types of beads, so very diverse in nature, especially when we compare that to sugar only had 3 different types of monosaccharides, and proteins have 20 different types. So we'll use both facilitated diffusion and active transport depending on what type of bead you have or depending on what type of amino acid you have. Most of them are going to use active transport. And most of it will take place again in the small intestines, duodenum and jejunum. And actually final protein digestion will take place after the absorption process, okay? So within the cell, the amino acid chains can be broken down into single amino acids, okay? So those amino acids enter the portal system and travel to the liver where they can be processed. And then, like we said, broken down into those single amino acid molecules. Again, just a review, what is active transport? How does this happen? So I'm going to move against my concentration gradient. Notice that I need an energy input in order to do so as I'm traveling those molecules upstream. Okay, so active transport will occur in order for me to move those short chain amino acid molecules across from the intestines into the bloodstream so that I can deliver them via the hepatic portal system to the liver so that they can be processed. Okay. Again, just a nice summary for you. If this isn't helpful, that's okay. We took that big long strand of proteins that was all kind of twisted up. We broke them down into large polypeptides, which are molecules with many proteins. Okay, poly means many. Then we broke it down into shorter peptides and then eventually into a single amino acid or single beads. Okay. Again, nothing new here, and if this is confusing, that's okay, just ignore it. Don't worry about these, we didn't talk about those enzymes in particular, but again, I'm gonna consume those dietary proteins. Once I get to the stomach, I'll have hydrochloric acid and pepsin involved in the digestive process. I'm gonna have denaturation of the protein molecules. As we move into the small intestines, again, I'm going to break those molecules into smaller peptides and amino acids. active transport will serve as our primary mechanism of movement from those molecules in the digestive tract to into the bloodstream, so that I can utilize them for a variety of things. So again, we mentioned that protein wasn't really that important for energy metabolism. So what do we need proteins for? What are we using these amino acids for? Okay, so once the amino acids enter the cell, they become building blocks for specific proteins. So again, now I have all of these beads available and I can use them to build whatever I might need to build within the body. Okay. Hormones may stimulate the production of particular proteins. Let's see. Testosterone can increase muscle size and thus increase the production of contractile proteins. So again, having a significant amount of testosterone will assist in building of different proteins. Also, DNA includes the actual instructions on how to construct a particular protein, and the DNA is located within the cell nucleus. Okay. We have this thing known as the amino acid pool, and the amino acid pool serves as kind of like this dynamic storage bin, think of it that way, of these beads, okay? And the amino acid pool consists of amino acids that can be found in the blood, the skeletal muscle, and in the liver, okay? And so if amino acids levels fall in one compartment, amino acids from another compartment can be mobilized to correct the imbalance. Okay? So these amino acids from the pool can be used for a variety of things. They can be used to synthesize new structural proteins, enzymes, hormones. They can be used to metabolized for energy. As I mentioned, this amino acid pool is dynamic and ongoing. So we're constantly pulling from one section and then replenishing depending on what foods we consume and what, what amounts of each of those protein, or what amounts of each of those amino acids we are consuming. If protein intake is not sufficient, protein from the muscle and other tissues can be catabolized or broken down in order to provide the appropriate amino acids to complete that particular task. Okay. We have essential and non-essential amino acids. So essential amino acids we have to get from our diet. Non-essential amino acids we can kind of like convert something into that. We can makeshift those amino acids within the body. Okay, so again, it's really important that we have this amino acid pool filled, and if I don't have the appropriate amino acids that I need to build whatever that structure is. Again, if it's non-essential, I can produce that or like makeshift that molecule from something that I already have. If it is essential, we can pull it from the amino acid pool, which we said was consistent of the blood, skeletal muscle, and liver. If the amino acid pool doesn't have it, then the building process cannot continue, and we have to wait to build that protein until we have that, which would come from a nutritional source. Okay, so here this kind of shows you how dynamic this is. So again, here you can see the gut, so digestion, I'm getting new amino acids. That's really the only place I can get them from. So then they will enter into this amino acid pool. I can use some in the form of energy. I will lose some in the form of urine and sweat. Again, I can use them to build within the tissue. I could also pull them from the tissue if I needed to, from our muscles, for example, to refill this amino acid pool if I'm not consuming the appropriate ones in appropriate quantities. Okay, so we've talked in a fair amount of detail about the 3 types of macronutrients. We also have two other types of nutrients that are important for us to consume and absorb, and those are our micronutrients, which are minerals and vitamins. Additionally, water is considered a nutrient of ours. Sometimes we see it put in with the micronutrients. I don't like when it's put in with the micronutrients because we need it in large quantities. But anyway, what happens with minerals, vitamins, and water. Okay, we don't need to actually break these down, so we don't necessarily need to digest these. But when food is broken down, vitamins and minerals are released within the small intestines. And most of those minerals are going to be absorbed in the small intestines. The large intestines will also contribute to absorption of certain minerals, including sodium, potassium, and chloride. Let's see. The process by which our minerals and vitamins will absorb is through passive diffusion. Fat-soluble vitamins can be absorbed with fat molecules. So basically, like they'll just move, they just like jump on board with the fat molecules and move across the membrane that way. Water-soluble vitamins and minerals will move through passive diffusion. And then calcium and magnesium can move through active transport. Okay. So as I mentioned, two different types of vitamins. We have water-soluble vitamins, fat-soluble vitamins. The fat-soluble vitamins will be carried with the fats in the micelles. They can be absorbed via passive diffusion, carried in the lymph if they are with a long-chain fatty acid. And then these can also be stored in our fat cells. Okay, water-soluble vitamins are a little more dynamic, we could say. They'll be absorbed with water in the small intestines. They're easy to move into the blood and they can move freely throughout the body. However, it's easy for us to excrete them because we can't necessarily store them. They're just kind of floating around. So it's almost more important for us to make sure we're consuming these water-soluble vitamins on a daily basis. Whereas the fat-soluble vitamins, we can store them somewhat, so not as essential that we're consuming them each day. Okay, just a few kind of concluding remarks as it relates to digestion and satiety, or how full you feel. And I wanted to kind of look at, how do we know when we're full, right? So how are these processes regulated? When do we know when to stop eating, right? So this is initiated, the feeling of satiety or fullness can be initiated by the stimuli within the GI tract or within the digestive tract. And there are 4 ways to stimulate those particular feelings. Okay, number 1 is distension or stretching of the walls. So again, that would have to do with the volume of the food or the fluid that we consume. Again, if I consume watermelon, for example, a lot of it, it's not a lot of calories. However, it is a lot of volume. So I might feel full faster because I'm eating like a couple cups of this and that fills up my stomach. Okay, so distension or the stretching of the walls. Number 2 is osmolarity or total solute concentration. So again, looking at the consistency of a fluid, for example. So if I consume I'm big into smoothies lately. I don't know if it's because it's summer or what the deal is, but you know, I'll make a smoothie with like banana and peanut butter and maybe like some coffee and a little bit of honey and some ice, and it's just like perfect, right? But it's very dense. Like there's a lot of calories in that. So its osmolarity is high and its calorie content is high. So that's going to make me feel full, even if I just consume like a 12-ounce cup worth, in comparison to if I just consume my 12-ounce cup of water, which has a low osmolarity. Okay, so again, looking at like the thickness or the consistency of that particular fluid would be important. Number 3 is acidity. Acidic foods actually fill us up. Okay, so spicy foods or acidic foods can make us feel full easier. So again, you maybe have seen before that people interested in weight loss should try to eat spicy foods. One of the reasons is because of the acidity of those particular molecules, or the acidity that we produce as a result of that. Probably the other part is like if I'm eating something spicy, I'm going to drink a bunch of water also, so that helps to stimulate distension as well. So something to think about. And then lastly, the digestive products. So how many or what's the composition as it relates to amino acids, carbohydrates, and fats. So again, I mentioned consuming a bunch of watermelon, for example, which is primarily just carbohydrates, but it's not very dense in calories. Like you can eat a full cup of watermelon and it's like 10 calories or something like that. If you imagine eating a full cup of like peanut butter, for example, okay, that'd be like thousands of calories. So even though the volume was kept constant, the composition was significantly different. So I would feel significantly more full with a cup of peanut butter in comparison to like a cup of watermelon. Stimuli can act on mechanoreceptors and chemoreceptors within the alimentary or the GI tract in order for us to understand what's going on as it relates to satiety. Okay. Lastly, there are two hormones that are going to play a role in determining how full or how hungry we feel. So we have leptin and ghrelin, and they're going to kind of work in opposition, okay? So leptin is released from adipose tissues, and it sends a signal to the brain to increase satiety or to make us feel full. So essentially, if I had a lot of fat, I should release a lot of leptin, which means I'm going to feel full off of less food than somebody who doesn't have a lot of fat, okay? If I don't have a lot of fat, I'm going to release less leptin, which means I need to eat more before I stimulate satiety or feelings of fullness, okay? Ghrelin is released from the stomach when the stomach is empty, and this will cause us to feel hungry. People that are on weight loss or type 2 diabetics, like their medication, will help to suppress the release of ghrelin so you don't feel hungry or you don't feel as hungry. And so the idea with that is that you can hopefully feel full faster because you're not hungry to start with. Again, that is it for the digestive tract. The next unit that we have, again, I know I have these listed as kind of the same lecture, and it's really two different lectures. So we have the digestive system and then also the renal system. So make sure you tune in to watch a little bit about renal physiology following the completion of this unit. Thanks.